Hello, everyone, and welcome to today's webinar, Advancing mRNA Therapeutics with Mass Photometry. My name is Louise Baskin of LabRoots, and I will be your moderator for today's event. This educational web seminar is presented by LabRoots and brought to you by Refine. To learn more about our sponsor, please visit refine.com. We encourage you to participate today by submitting any questions you may have during the presentation. To do so, simply type them into the Ask a Question box and click Submit. We will answer as many questions as we have time for at the end of the presentation. You may also use the Ask a Question box to let us know if you're having any trouble seeing or hearing the presentation. I'd now like to welcome our speaker for today's webinar. Mark Dickman is a professor of bioanalytical science and engineering at the University of Sheffield. He joined the Department of Chemical and Biological Engineering in 2003 and has established a dedicated RNA bioanalytical facility at the University of Sheffield for the characterization of mRNA medicines and oligonucleotide therapeutics. Professor Dickman, you may now begin your presentation.
Thank you very much for the introduction. Today I'm going to talk a little bit about some of our recent research at the University of Sheffield, where we've been utilizing mass photometry to characterize and analyze mRNA therapeutics. So just by way of introduction, my research group really focuses on the development and application of analytical techniques to characterize mRNA medicines and oligonucleotide therapeutics. So here at the University of Sheffield, we've essentially established a dedicated RNA analytical facility that houses a wide range of state-of-the-art analytical techniques. And we utilize these, we develop new assays to look at some of the emerging challenges in characterizing mRNA therapeutics. In particular, my research group, we've worked on bioseparations and biological mass spectrometry for over 20 years now. And these are really the two kind of main techniques that we use and we develop in my research group to characterize mRNA medicines. So we have a wide variety of different HPLC instruments in the facility, using different modes of separation, such as ion pair reverse phase chromatography, ion exchange, and size exclusion chromatography. And we interface these with biological mass spectrometry. We have a wide range of different high-end tandem mass spectrometry equipment to really give us powerful tools to analyze mRNA medicines. What I'm going to talk about a little bit more today is how we integrate alternative kind of solutions and analytical techniques, such as mass photometry, which has really emerged as a new powerful technique, which allows us to provide us very fast, rapid, sensitive analysis of mRNA medicines. So just a little bit of a background on mRNA medicines. And as you're probably all aware, the mRNA technology has really emerged over the last few years as a new class of therapeutic, a new class of drug. And this has really revolutionized the timescale required to develop new vaccines. And this has been exemplified by the Pfizer-BioNTech and the Moderna COVID-19 vaccines that have emerged over the last few years. But outside of vaccines, beyond vaccines, many of the big biopharmaceutical companies and many biotech companies have got a pipeline of new emerging RNA drugs that have potential treatments for infectious disease, cancer, cardiovascular conditions, and autoimmune diseases. These new drugs are really emerging and likely to come through over the next few years. So the expected market is really expected to grow predicted from around about $50 billion now to over $100 billion by 2029. So it's a really powerful new emerging technology. So I'm going to first talk a little bit about how we manufacture these mRNA medicines. It's in some respects a very simple system in how we manufacture or how we make these mRNA medicines. And this is done through an enzymatic synthesis. So here depicted on the left-hand side of this slide, we essentially just have an enzymatic process using the enzyme T7-RNA polymerase. It essentially copies the DNA template, uses the building blocks that make mRNA, the NTPs, to make our corresponding mRNA drug. However, one of the problems of this manufacturing process is RNA is quite unstable, so the purification storage is quite challenging. And through this manufacturing process, we generate a lot of impurities, such as things like double-stranded RNA, short mRNA fragments, or larger mRNA fragments, and multimers and aggregates. So we get a mixture of species that we generate during this manufacturing process, and it's really critical we're able to separate analyze and characterize these impurities. So if we look at the structure or the schematic illustration of what an mRNA medicine is, I'll walk you through some of the key different components and some of the challenges that bring us from a perspective of analyzing and characterizing these. So we have a wide range of what we call critical quality attributes, and these are key attributes that affect the efficacy of the mRNA drug, the stability, and the safety of these mRNA medicines. So you can see here on this schematic of what an mRNA drug consists of. At one end of the molecule, we have the 5' cap, and this really helps kind of translation and stability of the mRNA. We then have the 5' UTR, which again is involved in translation and stability of the mRNA drug. The coding sequence, so this is the sequence of the mRNA, which essentially encodes the antigen or the protein that's made from the mRNA molecule. And then at the other end of the molecule, we have this thing called the 3' A poly A tail, and this is just a constant or a sequence of A residues, which can be anywhere between about 50 and 200 nucleotides in length. So each of these different corresponding regions or attributes of the mRNA molecule, we really need to assess and characterize these using analytical techniques. So I've listed some of these below this schematic of an mRNA. So it's really important that we validate the identity, the sequence of our mRNA. Obviously, if you have the wrong sequence, we're going to generate the wrong protein and the wrong antigen. It's really important that the mRNA molecule, we measure the capping efficiency. We need to know how much is capped. We need to ensure we've got techniques that can address looking at the poly A tail length and the heterogeneity. And it's also really important that we have techniques and analytical techniques that can measure things like the integrity, how intact the molecule is. What are the impurities that are present? And just on the right-hand side of this slide, this is just a list taken from the latest USP guidelines. So this is kind of some of the guidelines that some of the things that we need to measure and some of the current techniques. So I've highlighted just a couple when I'm going to talk about in today's webinar, where we talk about the ability or the tools that are required to measure the integrity. In other words, how intact the mRNA molecule is. And one of the techniques where we look at purity, where we look at the presence of mRNA multimers or mRNA aggregates in the mRNA that's been manufactured. So having covered a lot of the critical quality attributes that we need to measure and monitor, I'm going to talk a little bit about some of the challenges in doing that. So first and foremost, that mRNA medicines are really large molecules. So these can be greater than 1,000 nucleotides in length. And actually, there are only limited current analytical techniques that are out there to really separate and analyze these mRNA medicines. And if we think about the mRNA itself, this is a really challenging molecule. It's really highly polar, negatively charged. With it being single-stranded in nature, it generates lots of dynamic secondary structures, which again leads to sample heterogeneity. And another big challenge that comes with characterizing and analyzing mRNA medicines is many of the impurities that we need to measure and monitor are very similar to the full-length mRNA. So they might only just be a little bit shorter, and this is depicted in this HPLC chromatogram here. So many of the impurities look very similar to the full-length mRNA molecule. And many of the abortive short transcripts are a lot smaller. And many of the impurities, such as double-stranded RNA, again, look very similar in nature. Very difficult to separate and analyze. And beyond these impurities, as we discussed before, we've got these critical quality attributes, things like the 5' cap and the 3' poly A tail, which also must be thoroughly characterized as part of any analytical procedure. What I'm going to do now is going to walk you through some of the analytical techniques that we've established in the group, some of the developments that we do to look at a number of these critical quality attributes. I'm going to first focus on the sizing of the mRNA molecule, and then talk about looking at the integrity, and then finally finish by looking at some of the recent work that we've done at the University of Sheffield, developing new techniques to look at mRNA multimerization. Okay, let's start with a bit of an introduction to mass photometry. As I said before, we're really going to showcase how we've integrated this now to really characterize mRNA medicines. So for those who are not too familiar with mass photometry, this is essentially a single particle technique. It operates in native buffer conditions, and it only requires very low amounts of RNA, nanograms of the biomolecule, and we can get information very rapidly in minutes. So here on the left-hand side of the slide, this just depicts the kind of the process in which mass spectrometry works. So essentially we have our RNA here in solution that we add essentially on top of a slide or onto a slide. So the slide is typically, in the case when we analyze nucleic acids, has a positively charged surface, and we simply just dilute RNA to appropriate concentration, which is very low. We add that to the slide and then put it into the mass spectrometry device. What the mass spectrometer does is essentially measure the mass spectrometry contrast, and that's the interference between the light reflected by the surface and the light scattered by the biomolecule. So that's what we're essentially measuring in the mass spectrometry instrument. And this mass spectrometry contrast essentially scales linearly with the mass or the length of the biomolecule. So this is depicted here on the right-hand slide. We measure this mass spectrometry contrast for hundreds, lots of different single particle measurements, and that allows us to fit histograms. And we get this nice correlation between the contrast and the mRNA length. So what we can essentially do is measure the counts, and this gives us information about the mRNA length of the particular molecule. So here's some examples of some of the kind of the typical RNA data that we get using the mass spectrometry instrument. So here's the first case where we're just looking at a standard RNA ladder that has between 200 and, or each of the markers or each of the RNA molecules is between 200 and 6,000 nucleotides in length. And we've got some of the mass spectrometry information that we get generated either using our own in-house polylysine slides or on some of Refine's mass class nucleic acid slides. Here's a typical example of some of the data that we get for the different RNA markers. So you can see here we now can measure the different signals for each of the different sizes of the different RNA markers, all the way here from 500 to 6,000 nucleotides in length. So all the data I'm going to show in today's webinar was all obtained on a Refine 2MP instrument and all generated using Acquire MP and Discover MP software. All the RNA data was typically obtained on the either in-house polylysine slides that we used or Refine's proprietary mass class nucleic acid slides. So we first did a little bit of benchmarking, look at our own slides that we've kind of coated in-house with the mass class slides, and we see very comparable performance between the two slides here for the RNA ladder. And here on the very right-hand side, now this is one of the mRNA medicines, one of the actual mRNA molecules. And again, we see we can get some nice information, rapid information on the mRNA and the sizing of this, which I'll talk about in a little bit more detail in the next few slides. Okay, so the first thing I'm going to talk about how we apply mass spectrometry and one of the big benefits of it, and this comes to the kind of the sizing of the mRNA medicine. So mRNA medicines can vary anywhere between over 1,000 to 6,000 nucleotides in length. And some of the really emerging new kind of self-amplifying mRNA medicines or self-amplifying RNAs can be beyond 10,000 nucleotides in length. This is a real challenge for us to kind of characterize and separate that. So traditionally in my research group, we've essentially done a lot of work and development using ion pair reverse phase chromatography. You can see here depicted on the left-hand side of the slide some of the high resolution separations that we can achieve, separating an RNA ladder from 200 all the way up to 6,000 nucleotides in length. So this is quite challenging to do. You need very dedicated stationary phases and columns, very high resolution systems to be able to achieve this separation. And this is an example using the common ion pair reagent called triethyl ammonium acetate, and we're able to achieve this kind of size-based separation. However, one of the problems or the big challenges when it comes to kind of analyzing mRNA using the same kind of ion pair reverse phase chromatography is depicted over here on the right-hand slide. So now on the right-hand side of the slide, I've got three examples of three different mRNAs: EGFP mRNA, N-look mRNA, and F-look mRNA. What we've got on the right-hand side is an overlay of these three different mRNA molecules now. And if we want to accurately size them based on the RNA ladder, you can see that the peaks really don't size very accurately using the chromatography. So for example, the EGFP mRNA is 931 nucleotides in length, but you can see it elutes after 1,000 nucleotide RNA fragment. And you can see even an even exemplified example here for the N-look mRNA. This is 871 nucleotides in length. It's actually shorter than the EGFP mRNA, but it elutes later than the EGFP mRNA and actually after the 2,000 nucleotide RNA marker. So in essence, ion pair reverse phase chromatography using something like a weak ion pair reagent is really not accurate in sizing your mRNA medicines. This is really due to the problems that the fact that the ion pair really separates not only on the size, but it also takes into account the sequence, the base composition, and the structure of the mRNA. So this is why we don't get accurate sizing using these weak ion pair reagents. You can move to more stronger ion pair reagents, such as dibutyl ammonium acetate or hexyl ammonium acetate. And this does give you more accurate sizing of your mRNA molecules. But the problem is separation of really large RNAs is really challenging using this approach. So in essence, just in summary, the sizing of mRNA using weak ion pair chromatography has lots of challenges. It's really not an accurate approach to look at the size of your mRNA molecule of interest. And also another common technique, capillary electrophoresis, and this is often used for looking at integrity and sizing of your mRNA molecule. And again, I'll talk you through some of the caveats and some of the data we've got, again showcasing that this technique also isn't the most accurate for sizing mRNA molecules. So in the top of the slide here, I've got an example looking at FLUC mRNA. This particular FLUC mRNA has 120 nucleotide poly-A tail. And then on the right-hand side, we've got it chemically modified. So a lot of these mRNA medicines often contain chemical modifications in them, such as N1-methyl pseudouridine. So when we put the chemical modifications in, obviously we don't change the size of the mRNA, it just has a different base in there. And if we do the sizing using the capillary electrophoresis, comparing it to an RNA ladder, again, it isn't that accurate. And again, you can see we get two different sizes, whether it's modified or unmodified. And in this case, both cases, the mass error was greater than 5%. So again, for EGFP mRNA using capillary electrophoresis, dependent on whether the EGFP is chemically modified or not, we get two different answers dependent on the sizing from this data. And again, the mass accuracy for these two examples, again, was above 5%. So when it comes to using mass photometry, we essentially can use the RNA ladder, as we showed before, generate a calibration curve used based on that RNA ladder, and then analyze some of our mRNA molecules of interest. So there's the example for the RNA ladder, based on 500 nucleotide all the way up to 6,000 nucleotides in length, generate a calibration curve, and then apply our mRNA molecule of interest, so in this case, EGFP mRNA. And you can see here, post-analysis, we get very good mass error accuracy, in this case, both below 1%. Very accurate in accurately sizing our mRNA molecule of interest based on the mass photometry data, showing distinct advantages over traditional techniques such as capillary electrophoresis and ion pair reverse phase chromatography. On the next slide, I've got a couple more examples here where we've shown both the analysis of NLUC mRNA, which is both chemically modified and unmodified. And you can see again here we get very accurate sizing, in this case less than 2% mass error, and very little difference on the readout that we get in terms of the nucleotide length based on whether it's chemically modified or unmodified. And obviously in a perfect world, in a perfect solution, they would come out at exactly the same size because we're not changing the size of the mRNA molecule, we're just substituting in a modification. And finally, at the bottom here, I've got shown in green, I've got an example of the EGFP mRNA, again, a bit of a smaller one, again, showing less than 5% mass error. So again, we've got nice different examples here showing the accurate sizing that can be achieved using mass photometry and kind of the advantages. So some of the other work we've developed or we've worked on here at the University of Sheffield is just to overcome some of the caveats and limitations of using some of the standard commercial RNA ladders to do this type of analysis. So here on this slide, I've shown a typical commercial RNA ladder, which has fragments all the way from 200 to 6,000 nucleotides in length. And with these particular RNA sizing ladders, we have an equal mass of each of the different fragments. So in this case, there's 120 nanograms per microliter of each of the different fragments. But of course, when it comes to the mass photometry analysis, you can see here we get a kind of a decrease in signal intensity as we head towards the larger RNA fragments. And this is because each of the fragments has an equal mass And obviously, as we increase size, we're decreasing the concentration, or we're decreasing the corresponding number of moles of the larger fragments. And this gives us a lower signal intensity on the mass photometry instrument. So what we wanted to do is essentially develop our own set of RNA sizing standards that were not based on equal mass to be put into the mass photometry. So we synthesized essentially an mRNA ladder, a series of different fragments. And each of these different fragments essentially was a better or more realistic example of an mRNA. So we developed a wide range of different fragments, all of different sizes, and each of these had a 104 nucleotide poly A tail. So it's much more realistic in terms of an example for the RNA sizing. And then what we could do is essentially generate an equimolar mix now of the mRNA fragments, put that into the mass photometer, and this is what's seen on the right-hand side of the slide now, where we have essentially the signal intensity is pretty much maintains the same across all the different fragments, and we believe this was a better RNA standard to use in the mass photometry workflow. So here's an example now of using this mRNA sizing standard that we have at equimolar ratios. We essentially use this as the calibration curve, and then this allows us to analyze much larger mRNAs. In this example here on the right-hand slide shown in green, I've got an example of where we've used the mass photometry instrument in conjunction with our in-house developed mRNA sizing standards, and this has allowed us to get less than 1% mass error for an mRNA that's greater than 4,000 nucleotides in length. And this is the COVID spike mRNA. So just in summary, we've been able to improve the accuracy of mRNA sizing using the mass photometer by moving away from the kind of equimass commercial ladders by using our own developed mRNA sizing standard that we generate at an equimolar mix. Okay, so beyond sizing, another critical quality attribute that we need to look at and monitor when it comes to analyzing mRNA medicines is this idea of looking at the purity or the integrity of the mRNA. So this is typically done using denaturing techniques such as capillary electrophoresis or denaturing ion pair reverse phase chromatography. And what we essentially do is we measure the amount of the full length material versus the amount of degraded short length material, and we get a measure or a percentages of the mRNA intactness. So here's some particular example data where we've taken two different EGFP molecules, one on the top there shown in green, which was generated straight from the in vitro transcription reaction with no purification. Corresponding chromatogram shown on the right-hand side is the denaturing ion pair reverse phase chromatogram. And essentially you can see very small amounts of degraded RNA in there versus the full-length product. So if we take the area of the full-length product versus the area of that small amount of degraded mRNA to the left-hand side, that allows us to essentially calculate the percentage kind of impurity or purity that we've got there. And you can see in the first example, without purification, the ion pair reverse phase chromatography, we get a number of about 75% pure or 75% intact. Now we can take the same material through into the mass photometry instrument, and with this being a single molecule technique, it's very sensitive for looking at those smaller amounts of degraded material. One of the caveats of using chromatography is we have a wide range of different short fragments, so our signal intensity or the UV absorbance is spread across all those fragments, making it very difficult to detect. With mass photometry, we don't have that problem, and we get the benefits being a single molecule technique. So on the right-hand side, you can see here that mass photometry data, we're able to also detect and measure the amount of those short degraded material to look at RNA integrity. So if we look at the second example shown below, so this is the same mRNA molecule, but now purified using oligo-DT chromatography. You can see from the denaturing ion pair reverse phase chromatography that the short degraded mRNA species are barely detectable. We've only really got full-length material present, and that gave us about 93% purity or 93% integrity of that mRNA. And then when we analyze that material on the mass photometry, again, you can see that most of the material, again, is the full-length mRNA molecule, with just, again, only short or very small amounts of that degraded material present. So here we've nicely shown that the mass photometry is a very sensitive technique to detect these short degraded material, to be able to measure the intactness or the integrity of the mRNA molecule. Okay, so in the final part of the presentation today, I'm just going to talk you through some of the recent work that we've looked at developing new and novel techniques to look at mRNA multimers or mRNA aggregates. So I'll first discuss what we mean by this mRNA multimerization and mRNA aggregation, and why it's a kind of a challenging critical quality attribute that we need to measure and monitor. So I've just got a schematic illustration showing what we mean by mRNA multimerization. So mRNA is a very large linear molecule. As we said before, it can fold up into a variety of different secondary structures, which is depicted here on the left-hand side. So this is just a single mRNA sequence, shown here as a kind of a static kind of secondary structure. So just one of these mRNA molecules or one of these units is what we call the mRNA molecule. Now in biology, lots of examples that two RNA molecules can come together and interact. And this leads to potentially where we have two of these molecules interacting together, this can form an mRNA dimer. So these can interact through a variety of different hydrogen bonding, base stacking interactions, things like kissing loop interactions, etc. So the whole variety of different types of interactions that stabilize these molecules coming together. And beyond dimers, additional RNA molecules can also bind. So where we have three molecules binding, this allows us to form a trimer and various other higher order structures. When lots of these mRNA molecules or mRNA monomers come together, this can lead to these mRNA multimers, which eventually can lead to things like aggregation and phase separation. So these mRNA higher order structures and these multimers, this can affect things like the translational efficiency of your molecule, and it might also potentially impact the efficiency of formulation into lipid nanoparticles. So these are considered critical quality attributes and something that we need to measure and monitor. So one of the typical most common techniques for analyzing mRNA multimerization or aggregation is size exclusion chromatography. This is essentially a form of chromatography that we can employ under native or non-denaturing conditions. And we're able to separate essentially the mRNA monomer from some of these higher order structures and species. So I've got two example chromatograms here shown at the bottom, where we're able to separate typically the mRNA monomer from some of these higher order structures. Well, some of the caveats of size exclusion chromatography is that you often have long run times, and you get low resolution separations. So you can see here, we can't get baseline separation of your mRNA monomer from your higher order structures. And obviously a lot of these size exclusion columns aren't particularly robust. So again, that's another caveat of this type of approach. So what we've been essentially working on is alternative techniques to analyze or characterize these mRNA multimers. So one of the problems of using techniques such as denaturing techniques like capillary electrophoresis or denaturing ion pair reverse phase chromatography is they're not suited to analyze and characterize mRNA multimers. So on the left-hand side of the slide, I've got two example chromatograms of the separation analysis of EGFP mRNA. And you can see we get the two main peaks of interest, but we don't see any additional higher order structures. They've been essentially denatured. But when it comes to analyzing these molecules using mass photometry, this is essentially a non-denaturing technique. It's operated at room temperature. So using mass photometry, we're now readily able to detect and see both the dimer and the trimer for EGFP mRNA in this case. So the analysis of these mRNA multimers, distinct advantages using mass photometry over typical denaturing techniques. So some recent work we undertook at the University of Sheffield was to kind of develop alternative chromatographic techniques to try and separate and stabilize these mRNA multimers as an alternative to kind of size exclusion chromatography. And we recently published this work in the Journal of Chromatography. Details of that recent publication is just highlighted here on the bottom of the slide, where there's more information. So essentially what we did in this piece of work was essentially look at altering or stabilizing the RNA-RNA interactions in the chromatography. So ion pair reverse phase chromatography, typically done using the ion pair reagent TAA. And you can see in the absence of magnesium, when we analyze EGFP mRNA across a wide variety of different temperatures from 40 to 60 degrees C, we see just a single peak. We don't see any higher order structures. So what we did to overcome that was we essentially included magnesium ions into the mobile phases. The magnesium ions are known to stabilize RNA-RNA interactions. And when we analyzed the same mRNA molecule under these new mobile phase conditions, you can see at the lower temperatures at 40 and 50 degrees C, we now see the presence of multiple peaks. And it was proposed that these new multiple peaks are essentially potential higher order structures, such as the dimers and the trimers and the different multimer forms. And then as we move to really higher temperatures, we essentially denature these and revert back to the mRNA monomer that's present. However, one of the caveats of this initial piece of work is whilst we've developed the chromatography, we've generated multiple peaks, we don't really know what these peaks are. So we needed to use alternative techniques to really validate and characterize these different peaks to confirm that they are the mRNA multimers that are present. So this is where the mass photometry came in. So essentially, I've just shown a schematic here now of the workflow that we've adopted in conjunction using the ion pair reverse phase chromatography, using magnesium ions to separate the different peaks. We then collected the individual peaks and fractions, and then we put them directly on the mass photometry device to look at measuring the size under native conditions of these different molecules. So essentially, we did the chromatography. We collected the different peaks, such as the peak 1, which we proposed to be the mRNA monomer, and peak 2 was proposed to be the different higher order structures, the different mRNA multis that are present. Collect those peaks, and again, do the mass photometry sizing analysis. So here's some of the data that we got, where we integrated the ion pair reverse phase chromatography, collected the peaks, and went directly to the mass photometry. So just to highlight, this is really one of the benefits that we see of the mass photometry device, is that due to the sensitivity, being a single molecule technique, we can take very small amounts that we generate from the chromatography directly through to the mass photometry. So we don't have to worry about low amounts, low concentrations, just simply take the peaks of interest and apply those directly to the mass photometry. So we essentially did the separations. Here we're doing the chromatography at 55 degrees C in the presence of magnesium. We generated the two different peaks, and we really wanted to just analyze those on the mass spectrometer to confirm that they are essentially the monomer and the multiple peaks. So here in red, we essentially captured peak 1, put it onto the mass spectrometry device, did the sizing, and it was shown to be the mRNA monomer with very little or hardly any dimer present, less than 2%. When we took the second peak that appeared on the right-hand side of the chromatography, when we collected that, put that through to the mass spectrometer and did the sizing analysis, again, it showed here very nicely that this peak corresponds to the dimer, or the presence of the dimer. Small amounts of the trimer are also observed. And again, we get some of the monomer species, potentially some of the equilibrium or degradation, or depending on how we've collected the fractions. So this is the first example really where we've been able to use the mass spectrometer to really show that the chromatography is separating the monomer from the multimer peaks. And we just got a couple of other examples where we did analyze some different types of mRNA, some different sequences, some different lengths of mRNA. So I've got two different examples here where we did exactly the same workflow. Used the ion pair reverse phase chromatography in the presence of magnesium ions to separate the proposed monomer from the higher order species. Collected the fractions, went directly to the mass spectrometer, which was allowed us to really confirm that peak 1, in this case, was the mRNA monomer. We didn't see any dimer or multimer present. And the peaks to the right-hand side are the higher order species, where we were able to observe the presence of dimer, trimer, and tetramer species in the mass spectrometer. So again, this really highlights the power and the advantages of using the mass spectrometry and combining it with the sensitive separations that we're also able to achieve. So finally, I'd just like to finish with some more recent work that we've done. We've really done a lot more optimization now of the chromatographic separations. We've scaled these up to preparative scale ion pair reverse phase chromatography. So we're now moving away to doing the separation on an ACTA HPLC using a preparative DNA pack RP column. So we're able to all scale this up, be able to inject and analyze over mgs quantities of mRNA, capture the mRNA fractions, such as the monomer, the dimer, and the multimer species. We're able to now desalt them, which is really important as well, because when we do the separations, we've got magnesium ions and ion pair reagent that we need to remove, because the magnesium can cause degradation in long-term storage. We can essentially preparatively do fractionation, desalting to remove the magnesium and the ion pair reagent, and then the mass photometry analysis to really validate and characterize the different species that we've collected. So here's the data from some recent runs on the preparative scale purifications. And you can see peak 1, shown here in red. We're able to validate on a mass photometry, only contains the monomer fraction. Peak 2 is essentially purifying the dimer fraction. And then peak 3, shown in purple, this is where we get abundance or a range of different multimers, as evidenced by the mass photometry data, showing all the way from dimers up to pentamers and hexamers that are present in these fractions. So this is really exciting. Now we've obviously got a system set up in place now where we can purify these different species, and this leads to lots of options and lots of different experiments that we're undertaking at the moment to characterize these and analyze the effects of looking at the different monomer, dimer, and trimer. Okay, so just to wrap up in conclusions, hopefully I've been able to show that mass spectrometry really offers a powerful tool for characterizing a wide range of different critical quality attributes of mRNA. First and foremost, it really allows us to do accurate sizing of the mRNA, less than 5% error. So this has real advantages over some of the traditional techniques that we've used in the lab, such as capillary electrophoresis and ion pair reverse phase chromatography. So sizing in these techniques can be influenced by chemical modifications that are present or the sequence and structure of the mRNA molecule. And it's also really challenging to get sizing for large RNAs, such as the self-amplifying RNAs that are over 10,000 nucleotides in length. Mass spectrometry can also offer opportunities to look at mRNA integrity analysis. So we get the advantages of this being a single molecule technique, and it's very sensitive to detect these short RNA fragments. So again, these typically can be very challenging to do. If you have a wide range of different small size fragments, our UV signal is split across these. So it can be very difficult to detect small amounts of a wide range of different fragments. Whereas the limitations or the caveats using mass spectrometry, we don't have that, and it's very sensitive at detecting these shorter mRNA fragments. And finally, I've shown that hopefully that mass spectrometry is a real powerful tool for looking at mRNA multimers and mRNA aggregates. So just to highlight, it's a non-denaturing technique, and we can do it in native buffers such as PBS. And we can also really show that it's really well suited to hyphenate with chromatographic techniques. So showing you many examples where we can do the chromatography, we can separate our peaks, our fractions of interest, and then directly validate and characterize those on the mass spectrometry technique. For being single molecule, again, we don't have to worry about low concentrations that we have or low amounts that we've got present. So in essence, we think mass spectrometry is a real powerful orthogonal method. It provides lots of additional insight compared to traditional tools such as chromatography and capillary electrophoresis, allowing us to get really accurate sizing, information on integrity, and analysis of mRNA multimerization. So just to wrap up, essentially we've established at the University of Sheffield a kind of a dedicated analytical facility where we have a whole variety of different assays and techniques in place to look at things like mRNA content, looking at sequencing the identity of the mRNA using techniques such as liquid chromatography and mass spectrometry. We use a variety of techniques, including mass spectrometry now, to look at integrity and the purity of the mRNA molecule. So we have a whole suite of different assays set up where we work with both a lot of academic partners, a lot of different companies, to analyze and characterize their mRNAs of interest. So if anyone's interested in working with us, then please do just drop us an email if you want to develop new assays or use our existing assays to characterize your mRNA molecules of interest. And finally, I'd just like to finish by acknowledging the students who did a lot of the work on the mass spectrometry and the chromatography in the group. In particular, I'd like to thank Alex Webb, who's done most of the work on the mass spectrometry, including all the recent work on the mRNA multimerization, developing the novel ion pair method and integrating that with the mass spectrometry work. And in addition, I'd like to thank Adam Meyer, Gareth Owen, and Sebastian Ross, who also developed the different mass spectrometry kind of analyses and workflows, including the new mRNA sizing standards that we use in conjunction with the mass spectrometer. With that, I'd like to thank you for your attention, and I'm excited to join the question and answer session, and I'm happy to answer any questions that you might have.
Thank you, Professor Dickman, for that presentation. That was a lot of really great information. We will now start the live Q&A portion of the webinar. If you have a question you would like to ask, if you haven't already, you may still do so now. Just type your question into the Ask a Question box and click Submit. So let's go ahead and get started. Our first question came in just a few minutes ago, so near the end of your presentation. Does the 5% error seem high, or can this become much lower in the future?
Okay, thanks for the question.
Not exactly sure which slide that might have been pertaining to.
Yeah, I think we're talking about just kind of summarizing some of the kind of the mass errors on the accurate sizing. So in a lot of the cases, we were able to achieve less than 1% error, depending on the type of mRNA that we're particularly analyzing. So for most of our applications, we were able to achieve, less than 1%. So I think 5% perhaps is on the high side. I think if we can compare to some of the orthogonal methods, I think that shows we often achieve 5% or more than 5% error, mainly because of some of the issues and the caveats associated with those other methods, such as ion pair reverse phase, etc. So from utilizing the mass spectrometry, typically we've been able to achieve less than 1% mass error. I think, just a couple of extra things to take into account that, these mRNAs aren't a distinct exact size. They have a poly A tail with a bit of a distribution of lengths. So that will also kind of potentially contribute.
Thank you. In the aggregate study, could you comment on the stability of the aggregate structures? Specifically, were the fractions measured immediately after fractionation?
Okay, that's a really good question. I probably didn't have time to go into the detail during the presentation. So what we've essentially done is, using the ion pair reverse phase method, especially the preparative one, we've been able to essentially isolate the fractions, and we've done both immediate analysis using the mass spectrometry to look at the different kind of populations. And what we've also been able to do is now, in combination with the desalting method that we've optimized, we've been able to essentially desalt, store at -80, and reanalyze over a period of time. And under the final optimized conditions we've got for purification of these different populations of the dimers, the monomers, and the multimers, we've been able to show that it's pretty consistent and stable. So we can now essentially purify them, desalt them, place them at -80. And over the limited time period we've seen so far, we've not seen any change in the population.
Question. How much variability do you see between in-house poly-L-lysine and mass glass?
Okay, so we only did some kind of preliminary data early on comparing the kind of the polylysine and the mass glass. In summary, just based on, small kind of sample size, it wasn't a completely comprehensive study, but we did look at the RNA ladders, some calibrants, and measure the kind of the unbinding events. And we saw very similar kind of data between the two slides. So in principle, it was pretty consistent. I would say there's a caveat that it probably depends a little bit on your expertise and experience of in-house polylysine slides. I think in the early days when we were using the scene, we were getting a bit of variability ourselves on how we generated these, but once we've optimized those, we've got more consistent performance using our own in-house polylysine slides.
Excited to hear how you answer this next one. Excuse me. Can or will mass photometry work on the next step of mRNA therapeutics? That is the mRNA protein interaction, to potentially verify the functionality of the mRNA.
Okay, that's quite a tricky question to answer. So first and foremost, in terms of mass photometry, then it can certainly look at RNA protein interactions. Although I know many groups and refine themselves have got lots of application notes and data demonstrating the ability to study RNA protein interactions. It's not something we've specifically done in this case, but there is the potential to look at the mRNA therapeutic binding to associated proteins, if you wanted to go down that road. I'm not sure if the question's alluding to also thinking about looking at mRNA LNP and LNPs. Then again, there's opportunities to use mass photometry to do the analysis of the particles and mRNA LNPs as well, but it's not something we've studied.
Next question. Depending on the circumstances and the type of RNA, magnesium can cause changes in the RNA beyond separating multimers. Do you run with very low concentration of magnesium in your mobile phase to avoid this? And are there any detrimental interactions with ion pair or pH modulating buffers like EDTA?
Yeah, it's a good question. I think there's a few different components in that one. First and foremost, we're essentially using the magnesium to essentially stabilize the RNA-RNA interactions. But the question's right that the magnesium can cause various different effects. Essentially, we chose to use 1 millimolar magnesium because that essentially represents physiological free magnesium concentration. So we're using at or around what the physiological concentration in the cell is. And obviously, it's important if we use too high magnesium concentration, then that starts to cause potential degradation of the RNA. So again, there's a bit of a balance. There's quite a lot of data in the literature kind of optimizing that concentration to use. We chose to use physiological magnesium. And more importantly, when it comes to doing the separations and purification, it is important to remove the magnesium post the chromatography, because long-term storage of magnesium certainly causes degradation of the RNA.
Okay. All right, next question. Would IP RP HPLC followed by MP be a suitable method for detecting and quantifying alpha-synuclein in blood samples?
It's a tricky question, probably outside of my remit, that one. It really obviously
And that's a fair answer.
depends on the concentration of that particular protein, etc.
Fair enough. We have a few what I'll call kind of application questions. What size of mRNA can be analyzed using mass photometry?
Okay, so I think this is one of the particular strengths of mass photometry in terms of the size of the RNA in which you can analyze. So it's really challenging to separate and analyze RNAs above 4,000 using ion pair chromatography or capillary electrophoresis. So mass photometry has the advantages to be able to accurately size or certainly analyze large RNA species well beyond 4,000. So we've been able to analyze self-amplifying RNAs up to about 10,000 nucleotides in length, and certainly also size mRNAs. The resolution issues are down more at the small size things below 500, but of course most mRNA therapeutics are above that size range. So I think it's one of the particular advantages of mass photometry is the ability to analyze a wide range of size of different RNAs.
Kind of along the same lines, what concentration of RNA do you need for mass photometry analysis?
Again, with mass photometry being essentially a single molecule detection system, this is again one of the particular strengths is that we require very low amounts of sample. So typically we're utilizing or using around about 1 to 3 nanograms per microliter of RNA, obviously depending on the particular size, but we're broadly in that range. So very low amounts, 1 to 3 nanograms per microliter has kind of been optimal for us. And you probably compare that with the chromatographic methods where we may require 50, 100 nanograms of kind of material.
Okay. And so again, similar question about concentration. So what concentration, range, and load volume or time works best to minimize surface crowding and artifacts?
Yeah, so again, it's kind of similar to that. I think within that broad range of low concentrations, you don't want too high concentrations because of crowding and artifacts, and you really want to minimize those unbinding events. But obviously, too low a concentration, you don't get the kind of number of counts or the kind of the single particle analysis. So there is a balance. You don't want to go too high, and certainly in any lower, and you just don't get this kind of the sensitivity.
Okay. Where does mass photometry add orthogonal value versus fragment analyzer or CDMS? So what might it reveal that others miss?
Okay, so it's a good question. I think this is one of the particular advantages. I think it's really a powerful orthogonal approach to the alternative kind of separation systems that are out there. So some of the particular strengths is, first and foremost, it can be done in essentially native conditions, non-denaturing conditions. So it readily allows you to look at the kind of the multimers and the aggregates under those conditions. Typically, capillary electrophoresis and ion pair chromatography is essentially a denaturing technique, so it doesn't have the power and the ability to do that. And then one of the other advantages of the orthogonality is the kind of the analysis of the integrity. So it's routinely we use capillary electrophoresis to measure the integrity, how much of the full length mRNA we have compared to the shortmers. One of the caveats of that approach is that the shortmers have generally low abundance and the signal intensity is spread over quite a broad range. So it's difficult to potentially measure those accurately. Again, with mass photometry, it's single molecule technique, and I think it has particular advantages of looking at those shortmers that may be present that you may miss in kind of capillary electrophoresis or ion pair reverse phase conditions.
And I think as part of that answer, you answered another one that's here, is the advantages. What are the advantages of mass photometry for the analysis of mRNA multimers or aggregates? And I think you addressed that a bit in terms of the denaturing capability.
Yeah, I think, it goes to foremost that it's a native technique, can be operated under essentially non-denaturing conditions, allowing you to analyze and potentially quantify the multimers that are present.
Okay. And I apologize, you may have addressed this in the magnesium question, but it's asking what buffers, salts, and additives are compatible with mass photometry for intact mRNA.
Okay, so I think that this kind of could be
Suggesting magnesium or mild detergents.
Yeah, so I think, again, one of the advantages that we see in the application of the mass photometry system is the kind of the broad range of buffers and reagents that you could use. So, in some respects, it's kind of matrix independent. So you can do the analysis in 1 times PBS. We've actually even done the analysis in ion pair reverse phase buffer by just taking the material straight from our chromatography. Don't even have to dilute in PBS and we'll still get good data. Or you can add magnesium to the analysis, or even doing it into detergents, moving to more denaturing conditions if you want by doing the analysis in urea as well. So a broad range of kind of buffers are compatible.
Okay, and we have one last question. How do you perform contrast mass calibration for RNA, and what standards do you use?
Okay, so I covered that a little bit in the presentation, but essentially we use an RNA ladder of known molecular weights or known sizes, traditionally ranging from, say, 500 nucleotides all the way up to 6,000. So we essentially use that as a calibrant to generate a calibration curve, and then we apply that calibration curve to our mRNAs of interest that we've analysed. As I said before, there are some caveats that we've developed and worked around in, most traditional commercial rulers are kind of have equi mass, which gives you a bit low intensity of the larger molecular weight or the larger size fragments. So we actually now have generated an in-house RNA standard where they're equimolar and covering a kind of a bigger mass range. And also what they are, each of those mRNA standards also has a poly A tail. So it's a bit more very similar to the kind of the RNA that we're analyzing. So it's essentially just, using a calibrant, a calibration curve. I would add it's also good to have an in-house mRNA standard that's very well characterized of a known particular size that you've analyzed by an orthogonal method, and then apply that to the calibration curve just to make sure you're within a certain mass accuracy. And then when it comes to analyzing your unknown mRNAs, you've got a good confidence that everything's set up correctly.
Perfect sense. Well, thank you again, Professor Dickman, for the presentation and for such thorough answers to those insightful questions. I also want to thank LabRoots for hosting this event and to Refined for underwriting today's educational webcast. Before we go, I also want to thank the audience for joining us today and for all of their insightful questions. The ones that I guess we had time for all of them today, but for any that get submitted during the on-demand viewing period will be addressed by the speaker using the contact information you provided at the time of registration. LabRoots will alert you by email when this webcast is available for replay. We certainly encourage you to share that email, share that link with any colleagues who may have missed today's live event and might like to view it on demand. So until then, next time, thank you again. Goodbye and have a great day.